that's, it shows very bad priorities, in my opinion. Giving up this talk to spend time on the beach in Puerto Rico. But nonetheless, let's go ahead and get started. Now, I have to say, most of my talks have always been on federal regulations and company procedures. This is the first talk I've given where the news changes almost minute by minute. So, as you well know, 3-part series. This is week 3, vaccines. And I'm going to be making references to the other 2 talks to bring it all together. We're going to start off with talking about the history of vaccines and some unique drug development issues with vaccines. And there are a number of them, and we'll talk about those. I'm going to go into the types of vaccines because it turns out there are 4 or 5 different types of vaccines. And then I'll focus on the SARS-CoV-2 vaccine. We'll focus on the current vaccines that are available, the variant issue, the pause that's happening and what that means. So first we have a pop quiz. Who is Edward Jenner? And you can type into chat just A, B, C, D, or E.
D. D.
Okay, and can I ask you all to stay on mute during the presentation? Because sometimes there's distracting noises.
Oh, sorry.
That's okay. Okay, so it looks like people are saying, everybody's saying D, except for Lon, he says A. So let's see what the answer is. Now I have to say, the Olympic gold winning athlete is actually Bruce Jenner. And the actress and TV personality is still Bruce Jenner, but it's after he transitioned to Caitlyn. The entertainment manager in number C is actually Kris Jenner. And then E is Jenner Furst, who is a movie producer. The answer we were looking for, of course, was the person who administered the first vaccine. And we are going to take a look at, these are the 22 diseases that are currently treated with vaccines. And I actually did not put up COVID here. So that's actually 23. But a couple of things I want to mention about these. First of all, interestingly enough, smallpox, the last case was in Texas in 1949. So it really wasn't that long ago. And I want to mention briefly adenoviruses, because I'm going to be talking about that a little bit later. But this is a virus that affects the military when they're in combat training. And this was very prevalent in the 50s, and there was a vaccine that was created for that, and it was used for decades. And the reason I bring this up, we'll find out later. But the point of this is I wanted to say that a lot of these diseases don't even sound familiar, and that's because they're no longer a part of our world. But they're still present in the developing countries, and it was very, very present in the 1900s. It was a different story then. And before I go into detail, I just want to have this pop quiz. What was the annual death rate of measles during the 1900s? And I'm going to try to drill down to what life was like in those days. So A, B, C, or D? And if you don't know, just go ahead and take a guess. Okay, so I see B, D, C. So the answers are all over the place. And let's take a look at what it was. It was more than 500,000. So that's more than 500,000 deaths annually in the US during the 1900s. Now, what's interesting is the population was much smaller then. It was actually in 1950, there were 100 million, only 100 million people in the US. So that's less than half of what we currently have. So imagine a disease that every year killed 500,000 people. And you get an idea of what life was like with these diseases. And another quick glimpse of what the world was like. This was the annual death rate of these what are now preventable diseases. So you can see, and this is where we see the measles was 530,000 people annually. And you see that most of these have been wiped out completely by vaccines. Mumps and pertussis, which is actually whooping cough. We've had some reported cases in 2016, and that's because people have gotten lax about getting their children vaccinated. And I don't even know if you remember, but in 2016, there were a number of whooping cough outbreaks. All right. Just making sure I covered everything. Now, I'm going to talk a little bit about the history of smallpox. I will also talk about polio, because smallpox was our first major vanquishing of a disease, and and polio has some lessons for us to learn. So smallpox, it was basically 30% of the people who got it died, and there was awful scarring involved all over your body, mostly your face. And it created these rashes, which they've actually found on mummies, which suggests that it's been around for 3,000 years. So the way it was treated was this process called variolation, and it was the variola virus. And what they would do is they would take the pus from a rash, from a blister that was occurring, a smallpox blister, and they would scratch it into the arm of individuals. And that would give them the disease, but it was often less severe. And so people recovered from it, and then they were vaccinated, or they would not get the disease again. Unfortunately, people did die doing that because it was actually the smallpox pus that was being put in. Now, what happened was an improvement. Edward Jenner had a major improvement. And what he noticed was that milkmaids who milked cows would get the cowpox virus. They would get cowpox, which was not fatal, but they would not get smallpox. And so he thought that maybe there was a correlation there, and that maybe if he gave the cowpox pus and put that on people's arms, that might solve the problem. Sure enough, he gives it to a 9-year-old boy. He takes from one of the milkmaids, he takes the pus, puts it on the arm of the 9-year-old boy, and lo and behold, he exposes the little boy to smallpox on 3 or 4 different occasions. He doesn't come down with the disease. So they start this huge campaign to vaccinate people, and they did it by the hundreds and thousands. They would just put this into their skin, and that would protect them. So the last case was in 1978. The world was declared free of it on May 8th, 1980. So that was the horrible disease that was vanquished by vaccines. Now I'm going to talk a little bit about polio. Now, the first epidemic was in 1894. It was in Vermont. There were 18 deaths and 132 people who were paralyzed. New York City has an epidemic about 10 years later, and it's right around this time that they first realized that the disease is contagious. They didn't even know that. It took them 10 years to figure that out. In 1929, there's the iron lung. It's first used for those individuals. I don't think I need to go into that. But it kind of surprised me because I just didn't realize it had been around for so long. And from 1935 through the 50s, they were trying to figure out how they could create a vaccine for this illness. And in 1950, there was an attenuated oral vaccine, and I'll talk about what that means. It's a weakened virus, and that was given to 20 or 30 intellectually disabled children. Well, number one, it turned out to be effective, that it was effective in preventing the disease, but it was criticized by people. However, it was common at the time, and we know that from Module 1, how these kinds of things happen, these sort of what you might call moral lapses. But nonetheless, it was proved to be effective. And then in 1952, Salk promotes a killed virus. So now this is a killed virus that is will not give the disease, will not spread the disease. And in 1954, trials begin on this. And in 55, the vaccination program begins, and everybody's taking these vaccines. So what happens is, later in 1955, there is what's called the Cutter incident. And what happened was there were 29 cases of polio. And what they were realizing, there was this manufacturing plant, the Cutter plant. And so what they did is they paused the program. They took a look at what was happening, and they determined it was a production issue, and that what was happening was the plant was not fully killing the viruses. And so what they were doing is giving these weakened viruses to people, and some people were succumbing to it. So they figured out how to improve not improve it, but get Cutter to implement the procedure so that all of the viruses were killed. And then the program continues, and it was continued to be successful. So in 1959, the Sabin vaccine arrives, and that's a sugar cube versus an injection. And some of you may remember that. It's easier to dose, and because now you just have sugar cubes, you don't have to worry about all these syringes. It has a faster reaction because it goes directly into your system, and it's good in an epidemic. The problem is that it can revert to the virulent form, and that's because it's not a fully killed virus. So we have these two very effective viruses. One is an attenuated live, and one is a killed. And the world weighs the risks and benefits of it for decades. And obviously the sugar cube is easier to give out in developing countries and places where they may not have the infrastructure. The bottom line is, as you can see, 1994, the Americas are considered polio free. In 1997, there is a decision to revert to an all IPV program. So they decide in 1997 that they're only going to give out the injection to prevent people from inadvertently getting the disease. So we're going to refer to some of these lessons learned when we talk about the current COVID vaccines. And this is just an interesting story that I couldn't help but tell. So the year is 1961. Chicken eggs are used to grow viruses. They still are for some viruses. And you think flu, measles, mumps, there are a number of others. And there is a situation with the chickens. They have a chicken leukemia. And most chickens have it, and it's not really, at the time there was a fear that might spread to humans, especially with these vaccines using the egg yolks. And we now know this is not true, but in 1961 we didn't. So a virologist from Merck hears about a farm in California that has especially bred disease-free chickens. So he goes out to this farmer and he tries to talk him into selling him these chickens. Well, the farmer refuses to sell, and he spends a day there trying to talk him into it. He finally gives up and figures he's going to leave empty-handed. And then at the very end, there's a conversation that comes up, and it turns out the virologist is from Montana, and so is the farmer. So the farmer decides at that point, last minute, he sells all of his his disease-free chickens to the virologist for $1 a chicken. And these chickens are still being used today to create these vaccines. Okay, so now we're going to talk about some unique issues with vaccines. Vaccines are biological. We know this from module 2. This is the only way that we can get vaccines is by using live viruses, germs, whatever. And they come from the germs themselves, and they're used to teach your immune system to fight off certain germs or certain diseases. Now, they're given to healthy people, and this is unlike any other medication. There is no disease. They're not given to eliminate pain. They're not given to treat a disease or cure a disease. They are given to healthy people, sometimes who are at risk for getting the disease or having serious adverse events. So this is a huge issue. So that's why there's a lot of attention paid to adverse events with vaccines for this reason. There's also the concept of herd immunity that you don't have with typical pharmaceuticals. And that's the point where enough people are immune to the virus that it can no longer spread in the population. And I want to talk a little bit about herd immunity. So this is a graphic, and you see the color coding of the people who are susceptible. They're vaccinated or protected in some way. Maybe they've had the disease and they're no longer vulnerable. And the infection, the infected people. All right. So herd immunity is when a sufficient percentage of the population is immune, either by having the vaccine or by having had the disease that they've recovered from. Now, the issue with vaccines is that people not vaccinated are at risk for causing outbreaks. And some people can't be vaccinated. Sometimes they're too young, or they have health issues that prohibit it, or are poorly immunized from the vaccines that they've received. So if you add a number of people who just simply refuse, then you run the risk of the germ bouncing around the population, causing outbreaks, like you see in A. And once the immunity has been reached, herd immunity has been reached, then the whole population is protected, even those very few people who are too young or have health issues, and like you see in B. Now, the current timeline, or I should say the current timeline as of February 24th, when the New York Times had this in, and they reported this in February 24th. The timeline for herd immunity that's up in the blue range that you see. Now, the bottom red dotted line that's the line that indicates the people who are fully vaccinated. The upper line is the people who have been vaccinated and have had the disease and recovered. So you can see that total immunity, we should be approaching herd immunity by July or so. The problem is that this is such a fluid situation, because if we get more vaccines, that could accelerate the timeline. But if we get more numerous effective variants, that will decelerate the timeline. And the herd immunity, it varies for each disease, but it's been determined to be, I've seen estimates from 70 to 90% of the population needs to be immune before you reach herd immunity. The reason for that is each disease, it depends on how communicable they are. And so if you were following this, you may have realized that a year ago, they were talking about 50% herd immunity, and it's been creeping up. And it's now, I would say, closer to 85 to 90%. And the reason for that is they just simply didn't know how communicable it was. And when they found out it's as communicable as measles, which is considered a very communicable illness, that increases the percentage of people that have to be vaccinated or have recovered from the disease before you reach herd immunity. Okay, so now I'm going to briefly go into the different types of vaccines. So you have live attenuated, inactivated, subunit, recombinant, conjugate, polysaccharide, that's a mouthful, toxoid, and then lastly, DNA and mRNA. And I'm going to go through these one by one, taking a particular deep dive into the mRNA. So live attenuated, I talked about that before. That is when you have the germ that's been weakened, okay? And because it's so similar to the disease, the results are very strong and very long lasting. However, it is bad for those with weakened immune systems or those with long-term health issues because they are live viruses. And the examples we have here are I don't need to read those to you, you can read them. Now in the year 2000, measles was considered halted in the US. However, what has happened in those last few decades is that people have started to be vaccine hesitant. And because of that, we've had some outbreaks, which I'm sure you've heard about. Now we have the inactivated vaccines. That's where the vaccines are actually killed. So they're not as strong as live attenuated. So you do need to have boosters, like, and I think the annual flu shot that needs to be done. But there's no chance of getting the illness with those types of vaccines. Then we have this third type. And what this means basically, these types use specific piece of the germ, and that could be the protein or the sugar or the germ casing. And they're very specific and they give a strong response. And they can also be used on people with weakened immune systems and long-term health issues. Now I'm going to talk about the meningococcal disease because this is going to be relevant later when I talk about the COVID vaccines and the development of vaccines. So the meninges is the fluid surrounding the brain. And this disease, this meningococcal disease, is prevalent in crowded situations like classrooms, college dorms, and the military. It can be treated with antibiotics, but still about 10 to 14% of the people will die from this. And also of the survivors, 11 to 19% will suffer neurological damage, limb loss, hearing loss, a lot of side effects from this. So fortunately, there was a relatively recent vaccine that was created for this. I'm gonna say it was in the last 20 years and has been very effective and has solved the problem. Now we have toxoids, and those are, what we do is we use toxins that are produced by the germ. And we people develop immunity to that particular toxin rather than the germ. So this type definitely needs boosters. And I think of tetanus, that needs to be done every 10 years. Now we come to the mRNA and the DNA. And I'm going to spend a little bit of time on this because it's been on everybody's minds. So just to start off, scientists have been studying and working with mRNA vaccines for years. And think of SARS and MERS. They were working on this type of vaccine for those illnesses when they came out earlier this decade. Now, this model actually makes the proteins that trigger a response. It has a shorter manufacturing time, which has been incredibly helpful in this epidemic. And there is no live virus, so there's no risk of disease. It does not enter the DNA of the cell, so there's no risk of affecting your DNA. And I say all of these things because there have been all these rumors about these issues. Now, this model has been studied in Zika, flu, rabies, a number of other diseases. So this was a known entity by scientists when this pandemic started. And as soon as the relevant genetic information on COVID-19 was available, scientists started designing the mRNA instructions for cells to build the spike protein to help battle this disease. So for the population, it sounds like they just pulled it out of their hat, but actually this has been in understudy for decades. And the future of these types of both the DNA and mRNA is they're hoping that this technology may allow one vaccine for many diseases. And also research is ongoing in cancer. And they're hoping to use this technology to trigger the immune system to target specific cancer cells. And of course, once they do that, that will be a complete game changer for cancer treatment. All right, so here's a depiction of the mRNA. And you see at the top, the depiction of the gene. And what happens is, you see the DNA in the top row. The middle row is the mRNA, which is the bottom half of the DNA is that mRNA. And you can see they have two examples of where it's pulled down. And this mRNA is separated. And then what it does is it creates a protein. So different parts of the gene will create different proteins. All right, so that's the idea behind this technology. And what happens is, this is how your body responds to it. So the injection sends the mRNA into the cells, and it goes into your immune cells, and it gives those cells instructions to make this harmless spike protein. The cell then breaks down the instructions and gets rid of them so that it's no longer in your system. And then the cells then display the protein on their surface, and the immune cells will recognize them as foreign, build an immune response, and make antibodies. And you see that on the second half, the second figure. You see where it's creating the spike proteins and your body is creating the antibodies. Now, the second shot is important because what it does is it increases the number of T cells produced, and it further bumps up the antibodies. Now, I don't know about the Moderna vaccine, but the Pfizer vaccine, that second shot also increases the memory B cell. That's what it's called, the memory B cell. And that helps produce antibodies years later. And studies have shown that the memory B cells can create, can have a memory that lasts up to 9 decades. So it's pretty impressive. They think that's why young people tended to die in the pandemic in 1918, and they're thinking it's because the older people had this protection from an earlier exposure many decades prior to that. Okay, so now we're going to look at the current vaccines that are available. We have the Pfizer and Moderna, and we know, I am sure you know by now, that that's using the mRNA technology. Now, the AstraZeneca and the Johnson & Johnson they have a different method. And I'll just point out, in case you don't realize it, the AstraZeneca was never used in the US. Only the Johnson & Johnson one was used. And so I'm going to drill down on Johnson & Johnson, but just be aware they both use the same technology, which is, instead of using the mRNA to deliver the instructions, these vaccines use the disabled adenovirus. And that was the virus I talked about earlier that affects the GIs in military, in camp during the 50s. So they took that virus that they knew about, and they use that to deliver the instructions. So it enters the cells, but it cannot replicate. So again, there's no danger of getting the disease. And it goes to the nucleus where the instructions are replicated. All right. And so again, it's disabled. So you have no risk of getting the disease. It only requires one shot. It's kept at normal temperatures. And this is very important when it comes to countries who can't handle the deep infrastructure that's needed for these mRNA vaccines. All right. Now, this adenovirus technology isn't new. I mean, it's very new, but Johnson & Johnson did get approval for this type of vaccine for Ebola. And they got that in July of last year. And of course, we don't really hear about that because Ebola is not part of our world, but there is some history there behind it. And so now I'm going to talk about the pause that has happened. And there are issues with J&J. There's no doubt about it. And these were noted with AstraZeneca, I have to say, but we haven't focused on it in the US because J&J is the one that's being distributed. But basically, there have been 8, and I say events, they're deaths, 8 serious adverse events out of the 7.4 million doses in the US, and 222 deaths out of the 34 million worldwide. Now, the chances of getting it are extremely rare. And I calculated out that it's 1 in, and that is 5 zeros, 1 in 5 zeros, 108 chances of getting it in the US. And you're a little bit more at risk outside the US. It's 5 zeros, 653, okay? So to put this risk in perspective, to think about it, the way I looked at it is I wouldn't buy a lottery ticket if my chances were 1 in those numbers. But I also view it as like taking a plane. Now, air flight is one of the safest methods of transportation. The chances of anything happening are infinitesimal. However, if you're in a plane at 20,000 feet and it starts to go down, the results are catastrophic. And so that's the same way to look at this. So it is rare. But again, think about how it's being given to healthy people. And in this particular situation, the adverse events seem to occur in younger people. Now, these younger people are at less risk of getting COVID than older people in the older 50 and older crowd. So when you start looking at it that way, you start to realize that, maybe this risk benefit is, maybe we need to take a pause. All right. So I'm going to come back to the pause, but first I want to give you an idea about what, because this will help me talk about the pause. I'm going to talk about the intense reaction that occurs. So what happens is this particular vaccine activates the platelets, and it causes a very powerful misdirected response. The body basically goes haywire, creating these platelets. So they start to clot, and they clot pretty much all over the body, and where they clot can have dire consequences, can be fatal. And also the platelets in your blood, the platelet level in your blood actually decreases because they're all clumped together in these clots. All right. What makes it tricky is that the standard treatment for clotting is heparin. And what they found is the heparin actually made it worse. And they were unable to predict who was affected. There was no obvious reason why these individuals were becoming susceptible to this. So, given all of that, a pause made sense. All right, because it's intended to understand the adverse event. We need to understand what's going on here. We need to learn how to treat it. And we need to learn how to limit the exposure of people, try to figure out who's susceptible to it, and so who might not be appropriate for this type of vaccine. And so I just want you to reflect back on the polio vaccine, there was a pause there. They had 29 people who got ill, and it wasn't even necessarily deaths. It was just 29 people who got the disease, and they had a screeching halt. They figured out what happened, and then they continued. So what I have been researching is there are 2 studies that I read about. Trust me, there are studies going on all over the world to try to figure this out. This is all hands on deck to try to figure out what's going on here with this adverse event. But these 2 studies involve 16 patients. 11 patients were in Germany and 5 were in Norway. And what they determined is that they all had antibodies known to activate platelets. Okay, so now we might have a clue as to who these people are. Both recommended IV immune globulin as opposed to heparin. So now we might have a way to treat it. And what I would like to propose to you, because I've heard from a number of friends and relatives who have said that they thought that this was overblown, this pause was overblown. And what I would like to propose is that this is science in action. Things are happening. You're giving these vaccines to healthy people. Things are, there are deaths involved. Let's just take a pause and try to figure it out. And my guess is, and I've heard that I just read this morning that the EU has already decided they're going to continue with the shots. And my guess is that my prediction is that we will get some information on this, and very shortly we'll be given some guidance. And that's what the point of the pause was. Stop everything, figure out what's going on, and then educate the physicians so that they know how to treat it, how to predict it, that sort of thing. Okay, science in action. Now I want to talk about the vexing variants. Variants are any mutation in the gene is a variant. And actually, why are there so many with this one? It turns out that this is actually typical for these types of viruses. And think of the flu when you know it mutates so often that you have to switch up the flu vaccine every year. So what's happening is the germ is trying different things to increase its likelihood of survival. And most things don't work and they get discarded. But what happens is once in a while, something will be very successful. It will help increase its survival. And so it continues it continues with that particular variant. And geography plays a role too, because when you have different pockets of people, there might be a genetically distinct variants for that population. Okay, and we've seen that already with the examples that we have down here, the 4 examples, the 4 current variants. And these are variants of concern. And I will tell you that, as I said, these variants happen all the time. They become variants of concern when either it reinfects people who have had COVID, or it's more transmissible than the basic disease or the basic germ, or it causes more serious disease. And those are the 3 things that they look for. Now, so far, the vaccines do seem to mitigate the variants, but that could change with new variants arriving. The mRNA produces a lot of antibodies, and so that should provide adequate protection. And the third thing that is kind of reassuring is that if it doesn't stop you from getting COVID, it should stop you from being hospitalized or dying. And that seems to be the case from what they've observed. All right. So the only way, or the best way to deal with this, is to continue tracking the variants. That means having the money so that they can do the sequencing on these samples that they get from people getting COVID tests to determine if there are new variants, and if there are, to monitor them carefully. But that's why we have to maintain safety measures. We have to maintain social distancing and the masking and all of that, because basically less spread means fewer opportunities for the disease to spread and to mutate. Okay. Now we're going to get down to the thing that I think people are most interested in, which is the talking about how this vaccine got deployed so quickly. All right. And I want to go through the issues when we talk about the standard development. And then I'll do a timeline comparing a side-by-side comparison of standard development versus this vaccine, this disease. So remember the meningococcal vaccine, that was the antibody that that was the vaccine that was going to treat a disease that could be treated with antibiotics. And so what the company has to do is justify the creation. If we create this and people don't do it because they say, well, it'll just be treated with antibiotics, then is it worth it to generate a vaccine? Or the HPV that is given to young girls to prevent cervical cancer. And it was a question about acceptance because this is actually a sexually transmitted virus. And I actually had a friend in Florida who said there was no way she would give this to her daughter. And because the implication was, it means that, you know, whatever the implication was, there was no way she was going to, and she's in the healthcare industry, she wouldn't give it to her daughter. And I think there was a situation where, you know, it's sort of tainted because it's a sexually transmitted virus. So the companies have to hold focus groups. They have to make a determination. Will there be a demand? Will it be cost effective? Will we make our money back? In module 1, we learned about they studied the disease to determine the lock and key. And sometimes it can take a very long time to figure that out. It took 15 years for polio. This was done by the Chinese in December of 2019. They had the genetic code for the COVID, for the SARS-CoV virus in December of 2019. In module 2, we talked about how long it takes to conduct studies. They take years. It's phase 1, 2, 3, 4. Here it was, the studies were done concurrently and they were done very simply. And I'll get on to that, I'll discuss that on the next slide. We have to wait for approval. Sometimes it takes 6 months to 2 years. We learned about that in module 2, to await approval. Well, here they used this emergency use authorization, so that we don't have to wait until it's approved. We can use this EUA. The other thing that slows things down is companies will hold off manufacturing the product until they actually get the approval. It's very expensive to manufacture these drugs. It's very time consuming. They don't want to do it until they actually have the approval. All right. So what happens here is justify the creation, done. There's a worldwide demand for it. Cost effective, again, you have however many people, 9 billion people ready to buy your product. Determine the lock and key, that was done. And the cost effectiveness was also Operation Warp Speed made sure that that money was not an issue when it came to studying this and creating this vaccine. Determining the lock and key, that was done. As soon as the genome was completed, doctors started working on a vaccine then and there. And they realized that it was because it was so similar to the SARS and MERS, that once they had the genome, they were off to the races. Conducting clinical trials. These were simplified and concurrent. Usually there are dozens of studies to look at, and they look at different aspects of the drug and try to answer different questions. Here they did the phase 1 to make sure it was safe. And then the phase 2 and 3 were done almost concurrently. And here they were very simple. Is the drug safe? Is the drug effective? And that's all they looked at. And so will people who have been vaccinated, might they spread the disease? They don't know because they didn't study that. How long is a person vaccinated for? They didn't know that either, because again, simple, is it safe? Is it effective? Those kinds of things we can figure out later. And in fact, we are figuring that out now. There are studies to determine that as we go along, but that shows you how they could shave off a huge amount of time with that. Communication was faster and more frequent. The FDA works with a sponsor on a regular basis. And when you're developing a drug, it's like we have questions. Okay, let's schedule a meeting for next week. Here it was, I am sure there was a red phone going from the FDA to Moderna and the FDA to Pfizer, where they could speak as quickly as possible in real time, and all questions were answered, all guidance was given immediately, because it was all hands on deck. The approval wait time, there's this emergency use authorization, and I would like to just briefly read the definition of it. Is to facilitate the use of medical countermeasures during public health emergencies. Under an EUA, the FDA may allow the use of unapproved medical products or unapproved uses of medical products for serious or life-threatening conditions when there are no alternatives. It's as if this was written for this situation, right? I mean, it was like perfect. So I want to go back to this. We've looked at these the last two classes, and I just want to show you that you can see full on how the time was shortened. And we talked about in module 2 how there may be up to 5,000, maybe 10,000 volunteers who take these products. With these, they had 36,000 who were in these trials. So, and they did it quickly, efficiently. And now we are answering some of the questions in the what would normally be considered like phase 4. We're answering those questions now. Okay, so I have another quick pop quiz. According to the WHO, how many deaths have vaccines prevented from 2010 to 2015? And you could put your answer in chat. Okay, so I see D and C seem to be the answers of choice. And this is, I think I mentioned worldwide. And you're right, for those people who said D, they get the gold star. It is approximately 10 million, and that's in 5 years, which is kind of astounding. Now, I want to say in conclusion that I've heard people say vaccines save lives. I've seen little posters and little memes, but it's not the vaccines. You can have the most effective 100% vaccine. If it's still in the vial, it does no good. It's the vaccination. It's the jab in the arm that saves lives. Now, another quote here from another statistic from 2004 to 2014 in the US, 21 million hospitalizations have been prevented and 732,000 deaths have been prevented. Now, the unfortunate thing is that vaccines have become the victim of their own success, because people have forgotten how deadly measles was and how other diseases similar to that, how deadly they can be. And there have been deaths from whooping cough in the US. There are deaths from whooping cough and measles in developing countries every year, but we've sort of been saved by it because of our vaccine use. Well, we're starting to get vaccine hesitancy, and we are starting to get these outbreaks and these deaths due to these preventable diseases. So I want to mention to be an educated consumer. I am hoping that this talk has helped you kind of understand the disease and understand the treatment, and so that you can talk to people about herd immunity with confidence, and help you understand the headlines of the day and maybe why the pause isn't such a bad thing. Now, the last thing I want to end on is this may be the new normal. All right. I've spoken with scientists and I've read a number of articles about how humans have encroached upon every ecosystem on Earth. And it's in this increasing human interaction with animals and insects that causes these diseases. And so, like the 100-year storms that are now every decade, we may be looking at these 100-year pandemics occurring more frequently every 10 years or so. So that's our own moment to pause, I guess, today. But I would like to end by saying it's important to stay on top of these diseases, and vaccines are a big part of this. Science can save us if we allow it. And so I hope you guys have a much better appreciation for what goes into, or what has gone into and continues to go into, creating these vaccines. So that is my, let's see, that's my, I'm now finished. That's what I'm trying to say. And so if there are any questions, you guys are welcome to, we have 10 minutes to ask questions. We can stay later. I have no problem doing that. But I just need to get, I'm going to get a screenshot of the attendees for Shoshana. So feel free to, you can either write your questions in chat. We can talk about any of the modules that you want. I see Frank and Janet both have questions. So let's do Frank first. You can unmute yourself and ask your question, and then we'll move to Mario. It says Janet, but I believe it's Mario.
Yes.
So, okay, so Frank, feel free to, you have to unmute yourself.
Hello.
Hello.
It's Mario. I think I'm on. It's Mario for Janet.
Okay, yeah, that's fine. Go ahead, ask your question.
All right, I'm waiting for Frank though.
Oh, Frank is still muted, so he has to unmute. So go ahead and go first, and then Frank.
I'm kind of clear on the technology mRNA that created Pfizer, Moderna, but I'm not clear on what you call the technology that AstraZeneca and J&J.
Okay, so what they are using, you know, the thing is that with the mRNA, they use the mRNA to carry the instructions into the cell. And what happens is, instead of using mRNA, those two are using a disabled adenovirus. So it's just a different way of having the message sent into the body. It's a different vehicle to take the message into the body to have it create the antibodies.
And that's a disabled. What is that? What is it
called? An adenovirus.
An adenovirus. Okay.
Right. And
virus.
Yes, and it's a new technology. We don't have any other viruses that have been treated this way. The only one we have is Ebola, and that was in July of last year. So this is very new as well.
Okay, thank you.
Okay, and so I think Frank is asking if we know anything about the Russian vaccine, and I have not done any research on the Russian vaccine or any of the Chinese. I think the Indians have one. I focused basically on what's happening in America. So I apologize for that. Does anybody else have any questions? Oh, Deb says, when will these vaccines be approved? Are you talking about, or are you talking about all of them? My guess is that when the dust settles and the FDA has the time, because what goes into an approval of any product is they go through all the data, they visit the sites that conducted the studies, and they review the data at the site. It's a very lengthy process. And so my guess is that it could be quite a long time because the mRNA seems to be so effective and so few side effects. And they're spending all of their time right now. They're spending their time trying to figure out what to do with the J&J and AstraZeneca vaccines. So they're very busy doing that. And then they have a bunch of other drugs that are in the pipeline that need to be approved. So it's going to be a while. My guess is probably more than a year, because it's almost like an asterisk at this point that it's been approved. Okay, does anybody else have any questions?
Yeah, it's Ellis here. Hi.
Hi, Ellis.
Hi.
Thank you for joining.
No, thank you. It was quite illuminating. Because of the current success of the shortness of the clinical trial on the current COVID vaccines, what do you think the future impact will be on the length of other clinical
Ellis,
trials in the future?
Can I just stop you? I have to say, I realized I forgot to record this, and I meant to, because there's one person who couldn't attend. Okay. So I was thinking about that, and I wasn't focusing on your question. So now that I've realized that I forgot to record, please repeat your question.
Well, that's easy for you to say. Okay. So, yeah, because of the current success and length of time of the current vaccines clinical trials, what do you think the future impact will be for the future length of clinical trials as we go forward? Do you think it will still be as long? And
I think that's an excellent question. And I think in some ways there may be improvements made so that things can speed up again. And certainly the process has been streamlined in this case. The problem is that because it was such an emergency, it was all hands on deck in every capacity, from research to approval to manufacturing. So I think there will be some improvement in timelines, but I don't think you're going to see something like this. I don't think you're going to see a drug go from start to finish in a year. I don't think that's feasible, but you may see some streamlining, some lessons learned, essentially. So it's a good question.
Thank you.
Sure. Any other questions before we?
Yeah, one more quick one, if I may. I always get confused between if you have the antibodies and then if you've got the vaccine, and how either one of those or those combined relate to you still being able to catch, to get reinfected.
Okay, so with each disease, there is you get the disease and you get antibodies, and it protects you for a certain amount of time. The problem is we don't know how long that time is, and we don't know for COVID at all. We're learning as we go along. Okay, and so the question, I guess one thing I would like to say is, even though you've had COVID, you still do need to get the vaccines, and you still should get both of them. I know there have been issues. I know in the UK, they decided just to give one out to people to at least get some immunity. And those decisions are made out of desperation. They're not made, it's not the best decision. You're doing it because you're desperate. And so the best thing to do is get both shots. I can tell you also that the what we call the long haulers, the people who have long-term adverse events from the disease, And these can be all sorts of symptoms. And, massive fatigue, they may have achy joints, all sorts of things, loss of sense of smell, et cetera. Those people have been helped by the vaccine. So they have the antibodies, but especially after getting the second shot, their other symptoms just about disappeared. And so these were people who thought they might be living with this for years or a lifetime, and it was resolved. So clearly having the antibodies will protect you, but the best thing to do is to get the vaccines, especially with COVID.
Correct. But
Did that answer your question? I don't think it did. Sounds like it didn't.
just to follow, but if you have both, the variance is still a problem, correct?
Well, yes. I think if you have both, then you're probably better protected. There's probably better protection with having the disease and having recovered and having the natural antibodies, and then having the antibodies from the shot.
Right.
But we don't know. And with each variant, we just don't know how it's going to fall out. If there's one that is particularly deadly, that's why they're variants. They just vary, and there's no predicting. But hopefully the goal is worldwide to get everybody vaccinated as quickly as possible, which will decrease the outbreaks and decrease the chances of this causing more problems and getting hold in the population again.
Thank you.
Sure. Any other questions about anything, any of the modules? Okay, so I think I have the screenshots that I need for Shoshana. And if there are no other questions, I want to thank you guys so much for taking time out of your schedules. And I want you to have a good evening.
And
Thank you.
We want to thank you.
Okay. It's good to see you all, even though I really couldn't see you. It was good to talk to you.
But we could see you.
Right. Hi.
Thank you.
Right.
Bye-bye.
Bye.
Take care, everyone. Bye-bye.
Bye. Bye-bye.